our next speaker is Sydney Avena from the Maine Aquaculture Initiative Center, and she also is a master's student at the Darling Center, University of Maine. And she's going to speak about some work that is a collaborative effort between multiple hatcheries and multiple institutions on trying to understand where we are with larval production out of hatcheries.
Thanks, Mark. Hi, everybody. I'm Sydney Avena, and I'm here today on behalf of a bigger collaboration between several hatcheries as well as research institutions in Maine that are focusing around developing hatchery culture of the Atlantic sea scallop. Thanks. And while I'm the one up here who's kind of talking today, this really could not be possible without everyone on this slide who's making this happen. So big shout out to all my co-authors too. So as we know, why we're all here, the sea scallop is a very highly valuable seafood product in the industry. And it's an emerging industry in the aquaculture sector as in Maine as well. So right now, we have less than 10 active growers in Maine, but there is a lot of interest from aquaculture farmers in Maine into diversifying into sea scallops. And there's almost 20% of current experimental and standard leases that list sea scallops as an approved species. That data comes from the Department of Marine Resources. And currently, right now, the only source of seed is wild collected. And we got a great rundown on that process from Nate previously. So that's currently right now where all of our seed is coming from, is from the wild. But why are we interested in kind of creating these shellfish hatcheries for sea scallops specifically? And hatcheries have a great benefit because they're a closed and controlled system. So we can really focus in on finding out those variables that are really going to give us the best production. This also allows us to get more of a consistent year-round supply of seed. And there's also many benefits down the road. Once we get this kind of basic hatchery culture production down, we can start looking into lots of other different factors that can enhance sea scallop production for aquaculture specifically. But also when we're thinking about stock enhancement too, all the things that we can do with that. And other scallop species are being cultured around the world. Great presentation from France. So there's the king scallops down there, Pecten maximus, as well as bay scallops, which are more warm water species. But sea scallops are really tough to keep in a hatchery, as we've learned over these years, and that primarily comes down to their life cycle. They're a cold water species, as we know, and their larval phase is really long. So it takes about 28 days for them in the water column as larvae to reach the point of metamorphosis and to turn into petavelagers. And this really lengthy larval phase increases a lot of hatchery expenses. So right now in Maine, a lot of our hatcheries are set up for oyster aquaculture, and their larval phase is a lot shorter. So we're thinking about like a 2-week production period instead of the sea scallop, which can be up to 4 weeks. So this specific project is a 4-year collaboration between 3 hatcheries, which are both commercial and research hatcheries in Maine, as well as researchers at the University of Maine and our partners down in Connecticut at the Noah Milford Lab. And so this has been a really unique collaboration because we've been able to share a lot of our standard operating procedures as well as troubleshoot together to really get to the bottom of this production cycle. Each hatchery is focused on different larval experiments during the hatchery season, while we're also working towards kind of consensus protocols that we can all implement in our hatcheries to try and produce sea scallops. So in the next couple slides, I'm just going to go through a few of the methods that we've been using, similar to what Florian just did for his hatchery in France, just to show that what we've been up to, what we've discovered. And these methods pertain to the spawning of sea scallops, the larval rearing, and finally setting procedures. And again, we're trying to reach toward this goal where we can really develop these protocols for sea scallop production of spat in the hatchery so we can enhance our aquaculture industry as well as anything else we might want to do with lots of sea scallops. So the process of spawning, we take males and females. So unlike king scallops or bay scallops, these are different sexes. So we have male with the white gonad, females with the red. We spawn them using the thermal shock method. And interestingly enough, and I think Phoebe's going to speak on this in our next talk, we've definitely had more success spawning what we've been kind of calling like domesticated brood stock. So stock that has been raised in lantern nets versus brood stock that we've just collected from the wild. So we bring those into the lab. We place them in table, water bath, and we spawn them using a thermal shock. So we're just raising or lowering the water temperature a few degrees to really simulate spawning. And there's a little photo there of, video of a female sea scallop releasing eggs. And so we collect the eggs and fertilize them with some pooled sperm from several different males before stocking in our tanks. So we hold them for about 3 days undisturbed until metamorphosis is complete. The temperature should not get above 15 degrees Celsius. We typically see a low rate of conversion. I think that number has gone up in recent years as we've been getting a little bit better as we go along. But it's about 50% from eggs into D-stage villagers. And then we begin feeding at day 3 as well. So during the larval phase, we keep seawater filtered to 1 micron and the water temperature usually around 15 degrees. With really light aeration, we do buffer our cultures to a pH of 8.29 to 8.31. We don't do any sort of grading, which is separating out larvae by size, which is a pretty standard practice with other species, just because sea scallops are so sensitive to kind of any handling or any change in their environmental conditions. We feed a mixed microalgal diet to 30,000 cells per mil. And so after that larval phase, when they're beginning to show signs of setting, which is the development of a foot, and they also start to raft together during the Buckhead drain downs, we place setting materials in their tanks. So this only one hatchery in our collaboration has gotten this far at this point. That's Mook Sea Farm. Big shout out to them. But so far they've tried the methods of setting on a screen with an airlift, which is there on the left, and also the method of just setting on a spat bag, which is the same exact kind of process that you'd use in the wild collection of sea scallop spat. Just setting a spat bag in the tank filled with the Netron, that kind of blue netting material, and letting sea scallops settle on that. And we've seen better setting success with these spat bags compared to the screens. This is just kind of observational data at this point, but we have noticed there's been a lot more mortality on those screens as compared to the spat bags, which has been interesting. And so kind of alongside these methods that we're developing, we're also conducting little experiments as we go along to try and get at this big goal of what can we manipulate to really boost growth and survival, because we've just been seeing tons and tons of mortality as we go along. So the first experiments we did was static culture versus flow-through culture. We've also experimented with the effects of buffering and using different buffering compounds in the cultures. We've looked at different temperatures. Sea scallops have been cultured kind of in a range of temperatures from 12 to 17 degrees, which may not seem like that much of a difference. But if you're running a hatchery and you have to be chilling or warming the water to specific temperatures, even a few degrees can help save you money. So getting more information on exactly what kind of temperatures that we can really optimize. Tank size and shape. So there's some research out there that shows that larger tanks with less surface area will expose shellfish larvae to less of a bacterial load because tank surfaces have higher bacterial loads than a water column does. So continuing to experiment with that to see if there's a difference, as well as the effects of UV filtration for water culture, and continuing to experiment with diet. So we've had lots of different kind of parameters to keep looking at, and there are many more as we continue to look into this. So here's just a brief overview of the experiments that we've attempted so far since 2021 at our 3 different hatcheries. And so you can see that in some years we're repeating experiments across hatcheries or over years, and some that we're kind of just doing one-off. And these all have to do with the capacity of the hatcheries that we have at the moment, what their availability is, sorts of things like that. And the other thing to big thing to note here is that only one hatchery so far has been successful in producing set juveniles. That's Mooksy Farm. They've been successful 4 years in a row, producing sea scallop post-set larvae, which is excellent. It's really cool to see that happening. But the other two hatcheries, we've been really struggling with high mortality rates and culture crashes. So continuing to work through why that might be, what those differences are, and really nailing down our protocols as well. And so I just wanted to go through a few of the results of what we've learned through the years, and then kind of zoom out and do like a multi-year kind of what we've learned as well. So these are the results of static versus flow-through trials at Mooksy Farm that was conducted in 2021. So on the left there is percent survival, and the right is growth rate. And so this is split up into phase 1 and phase 2. I don't know, it might be too small for you guys to see that. I apologize. But on the left there, from larval days 3 to 9 is phase 1, where all the larvae were kept in static culture. And then for the later D stage to settlement, we split up into flow through and static culture. And so that kind of bright magenta pink color is the static culture numbers. And so you can see on the left, there was significant difference in survival between the static and the flow through cultures, as well as a significant increase in growth rate between static and flow through. So based on these results going forward, we've really focused on using static culture because that seems to make a big difference in larval growth and survival. So these are some of the buffering trials that Mooksy Farm did in 2022 and 2023. So they tested 4 different buffering treatments. In 2022, there was a no buffer control treatment tested against a soda ash buffer and a proprietary buffer compound that is used by Mooksy Farm to buffer the water in their oyster hatchery. And then in 2023, this experiment was repeated, but instead of that proprietary buffer, we used a soda ash and baking soda combination buffer. The growth data did not show any significant difference between treatments, so it's not shown here. This is just percent survival. But in 2022, there was a significant difference in survival using the proprietary buffer over the no buffer and soda ash treatments. But in 2023, we did not see any difference between buffering or no buffering treatments. So kind of some interesting results there. Still definitely things to keep working on. Kind of seeing how much of an effect buffering has versus no buffering, and the compounds that are used there too. So these were temperature trials that we did in 2023 at the Darling Marine Center. And so the percent survival data, everything ended up crashing in this trial. We didn't end up getting any post-set larvae out of it. But during the larval phase, we did see a significant difference in growth rate with temperature. So the green bar there on the right is 11 degrees Celsius, and we also tested 14 and 17 degrees. And the 11 degrees Celsius definitely had a slower growth rate and ended up with much smaller individuals at the end of the trial as compared to the 14 and 17 degree treatments. So continuing to increase our understanding of the effects of temperature on sea scallop larvae there. So now I wanted to zoom out a little bit and compare year to year the success that Mooksy Farm has had. So growth rates at all hatcheries have been increasing over the period, so which is pretty exciting. We are getting better as we continue. And this graph is specifically at Mooksy Farm, and the red bar there is the 2021 growth rate, and the blue bar on the top is 2024. And so each year, larvae are growing faster. They're getting bigger faster. In 2021, our growth rate was about 3.9 micrometers per day. And then up in 2024, that growth rate has increased to around 6 micrometers per day, which is a huge increase. And that really makes itself clear when we look at the first occurrences of eyed larvae present in the tanks. So that's typically referred to like the point of competency when most of the shellfish larvae in the tank are prepared to set. And that's when we add in our setting material. And so that point, that day in 2021 occurred at larval day 33. And in 2024, that day occurred at larval day 24. So that's a 9-day difference, which is really huge when you think about the costs of running a hatchery for over 30 days versus just over 20 days. So that was really interesting to see. And this increase in growth rate and the shortening of the larval phase also shows how much we've learned during this process, how much we've learned during the years and the skill building that's been taking place at our hatcheries. So this is looking at survival, specifically at Mook Sea Farm in 2022 through 2024. And the far right there, those three columns, kind of the end of the rainbow colors, show in 2024, the three treatments and the survival at the time of setting. So at day 24 in 2024, our average percent survival is around 70%, which is a huge increase from what we've been seeing in years past at that same kind of point in the larval cycle. So for comparison, in 2023, at day 28, we were around, looks like maybe 30% survival at that same point in the larval life cycle. So even though this is kind of just a snapshot of average percent survival, it is really exciting to see some of these numbers really start to come up production-wise. So in 2024, you're probably wondering, what are the numbers, right? So they approximate that they produced around 3.5 million settled spat in 2024, which is huge numbers. It's really exciting. And to kind of put that into perspective, it's settled on around 18 spat bags. And so our plan for these guys for right now is overwintering half of these spat bags in flow-through systems inside at the Darling Marine Center. That's what that picture on the left is, or really simple bait barrel systems just with flow-through water. And then we also ended up deploying half of those bags at a farm site, Pemaquid Oyster Farm site in Clark's Cove. And so the plan with those is to continue to track mortality rates kind of once a few months have gone by and they're a little bit bigger and easier to handle. Which brings us to some of the persisting challenges still to work through in this process. So kind of the big one, right, is the really high rates of larval mortality we've been seeing that result in culture crashes, mostly at the Darling Marine Center as well as the Down East Institute. And this has been kind of definitely a learning curve as we all learn to like work with this different species. But it's difficult when we aren't grading a species per se, because then it's really hard to separate dead larvae or dying larvae from healthy larvae in the cultures. And this also happens really fast. So like once we start to see mortality in a culture, it can happen, as quickly as maybe 12 or 24 hours later, then your entire culture is dead. And that's a lot of work, especially with a sea scallop culture that has been going for, 28, 30 days at that point. And finally, the system that we've been using, that we've been finding successful of settling on the spat bags, is really hard to estimate what we're coming up with at the end. Because they're setting on like a 3D surface and they don't set evenly, it's really hard to estimate what we're kind of getting out of those spat bags. So like I said, we think it's around 3.5 million that Mook Sea Farm was able to set this year, but that number is really kind of just a guesstimate. So working on methods to quantify set animals and maybe looking at different types of setting materials in future trials that are a little bit easier to quantify and work with will definitely help going forward. And something I didn't put on here, but Nate brought up too that I thought was really important is the transport question, right? So when these guys are coming out of the hatcheries, they're really tiny. They're set on, spat bags, Netron, and they're really fragile. So then kind of thinking about how do we transport those out to aquaculture farmer or into the water without killing them all is a big point of research that we still need to think about. Because right now they're just kind of going really short distances. Mook Sea Farm and Darling Marine Center are, probably 2 miles apart from each other. So it's really easy to get them there. But transporting over larger distances too will be something to work on. So thank you for listening. There's been a lot of really cool advances in this research in the past few years. We have a lot to learn, but it's been exciting to work on this project. So is there waving scallop? Thank you.
Thank you, Sydney. We have time for one question, and don't forget, we're going to have plenty of time for panel after a short break. So you have one question here.
Thank you. All the presenters, great presentations, a lot of information there. Can't wait to sit down with some of you and corner you. So I'm involved in the oyster industry for many years. And one of the things that we have done is kind of visually genetically select very large oysters to spawn. Something has allowed these oysters to live through multiple disease cycles, such as MSX and dermo, and why. And we have found great success in that. And I'm wondering if there was been any discussion about going out and finding the largest scallops you can find, and and then breeding those scallops to see if there's a different survival rate, because they're so susceptible at such a in their larval stage to anything. You sneeze and you just killed the whole freaking set. So thank you.
Yeah, that's a great point. And I think in the future, down the line, selective breeding is definitely something that's going to get a lot of attention, a lot of research. Right now, we're still kind of just trying to figure out how to do it. So just kind of using scallops that we have access to, as well as scallops that are farmed versus wild, for instance. So going out, finding like really large wild scallops versus scallops that have been produced on farms, or at least grown on farms. So yeah, I think that's definitely a big area of research that's going to happen in the next few years for sure.
Thanks, and I apologize. I don't think I introduced myself. I'm Peter Hughes. I'm with Atlantic Cape Fisheries down in Cape May, New Jersey. Thanks.